Good morning, and thank you for coming. Today, I'm going to be talking about open access to DNA. I'm going to start with a little bit of background science and talk about what interesting DNA evidence is, what investigative DNA databases are, and why it's good to have access to them. So, more than half of the DNA evidence that's collected these days is not obvious to interpret. There's uncertainty in how it can be interpreted, and most of it is made of low-level or mixed DNA. So I wanted to show you a picture of this. This is a DNA mixture. A DNA mixture, as you see, has more than 2 peaks. Ordinarily, if you look at data, you would see that there are 2 peaks, each corresponding to one of the parents and somebody's genetic type. And so, since people only have 2 chromosomes, right, paired chromosomes, 23 pairs of them, when you see more than 2 peaks, it means there could be more than, or there would be more than one person in there. Okay, so this is what the data look like. The real data is quantitative. Some peaks are taller, some peaks are shorter. The heights of data peaks correspond approximately to how much DNA is present, and so there's less DNA here at the lower peaks than there is on the right at the higher peaks. And the number is giving an indication of size on the x-axis, horizontal scale, and that's telling you what the value of that gene-like allele is. So if you have a pair of chromosomes at some genetic location, then you have 2 alleles. The alleles are just, in this case, those numbers, how long the molecules are. I'm not going to go into a lot of biology. I just want to see what data looks like. The reason it's good to know what data looks like is to get a sense of how it's interpreted. The goal is to figure out the genotypes that are present in the evidence. And for an individual, that's easy because there'll only be 1 or 2 peaks in the data corresponding to that person's 1 or 2 alleles. But when you have real data, most of it nowadays, and it's more complex, there's uncertainty because there are a lot of different possibilities. There could be one person who has 2 alleles, shown here in blue, with a lot of quantity, and another person with 2 alleles that are shown here in green in less quantity. So there are 2 people in this mixture, and that would be a way of explaining that data pattern, too low, too high. But there are actually a lot of possibilities, and mathematically you have to try out everything because there are really many, many explanations. The goal is to explain the peak pattern, and that's something that people really don't do at all well because it involves hundreds of possibilities at each locus. There are 15 loci, and people are not computers. If a computer spends a day thinking through 100,000 possibilities, studies have shown that a person can go through some of this in about 2 days, once, as opposed to 100,000 times. So for a complete review, you need computers to try to explain the data. And the result is, for one contributor at one location, is a reflection of what all those possible pairings were. Like 15, 16 would be that the minor contributor is the 2 green bars. But there are other possibilities. And what you end up with in the end is a listing of the possibilities, and because this is science, uncertainty is expressed as probability. So in this case, perhaps 90% of the probability goes to the minor contributor having those 2 green bars, that his genotype is a 15, 16. The reason you want the genotype is that after it's objectively inferred only from the data, never looking at a suspect, you can then make a comparison. Suppose you compare this with an individual that has a 15, 16. You can compute a statistic called likelihood ratio, and it turns out that the likelihood ratio is directly related to identification information. So any valid DNA match statistic is somehow related to a likelihood ratio. What we just saw is that at 15, 16, the evidence came in with a 90% probability at 15, 16. Suppose the probability of coincidentally seeing a 15, 16 pairing, that allele value in the that genotype value in the population was 9%. So if you took a random person from the room, they'd have a the chance that they'd have that 15, 16 is, say, 9%, 9 out of 100. That ratio of the evidence probability of 90%, that is a 15, 16, matching to some individual, divided by coincidence. In this case, 90 divided by 9 would be 10, and 10 would be the match statistic at this one of 15 loci. So for big matched numbers, multiply 10 times 10 times 10, you get a quadrillion or so. So after you've explained the data, you want the identification information, because these numbers get very big. Instead of trying to impress people with knowing what a quadrillion is or something, it turns out quadrillions are 1 followed by 15 zeros. Scientists express information in terms of the number of zeros. So 15 would be the information. In this case, there's 1 0 in 10, right? 10 is 1 followed by a 0. And so the logarithm, or the number of zeros in the number 10, is 1. Okay, so that's information value of 1. What's nice about it is you can add up information. If you get a factor of 10 at 15 different loci, each one has 1 0. So instead of multiplying them, you add 1 plus 1 plus 1. 15 times the final match statistic has 15 zeros in it. All right. Now, human review doesn't do all that. Instead, it determines a line, it draws a line, it asks what's over, what's under. If the line's too high, like if we're up here at 1,000, you throw out peaks. It doesn't even have to be give you the right answer. What it says instead is any peak that you see that's over that threshold is included, and that notion of inclusion means that all 10 of these allele pairs are possible. Even though looking at it, it obviously can't be right. That's what human review does, to simplify the problem into something that a person can do in half a day instead of spending a century on it without using a computer. And so we see that alternatives that don't look like they're right, a low peak, a high peak, end up with a lot of probability because it all adds up to 100%. So you're drawing probability away from the correct answer. If it reduces to 9%, then dividing 9% by 9% gives you a match statistic of 1, which means it could go either way. It's completely uninformative. There's no zeros after the 1 in the number 1. So that has 0 information. And that's a major source of what happens if DNA is locked up or owned by organizations that have a monopoly on being able to do the interpretation. Fortunately, there's discovery processes, other opinions, and so on. But about half of the mixture evidence currently, from studies that we've seen, is called inconclusive, when in fact most of the time it's not. And for the half that is given a match statistic, it's usually on average about 1 million times less than what the true information is. So there's a lot of information that's being lost. So we did a study with the local Allegheny County lab, which I presented at the American Academy meeting last month. And we looked at 2- and 3-person mixtures over 16 cases, wide range of offenses, 41 genotype matches. What we did is we looked at each locus. Their experiments have at most 15 different chromosome locations or loci when they get a DNA profile. And what we did is we looked at each one separately. And what we saw is we just measured that information, the number of zeros, right? So 1 would be a 10, 2 would be 100, -1 would be 1/10. We asked what was that information, the logarithm or the number of zeros over these over 500 loci. The answer is about 3/4 in sort of a bell curve. 3/4 means, what does 3/4 mean? If you had 16 loci, each of which giving you 16 loci, each one is giving you 3/4. 3/4 of 16 is 12. So on average, if you had 16 loci, you'd have 12 zeros, or a match statistic of about 1 trillion. That's what this means. But we're breaking it down, not for all the loci, but just one at a time. Okay? And so on average, that 3/4, or 0.75, means that the total match statistic would be around 1 trillion. So this is what normal data look like in science. Now, it's a bell curve. It's got variation. There's the standard deviation. What happened with people? Well, this is an interesting thing. This is the standard human review method in the US, combined probability of inclusion. But we're looking at it per locus, so it's really just the probability of inclusion. And what we see in the red bars is that it shifted to the left. See that? It shifted. It has the same width as a curve as the blue one, right? So the whole thing just kind of moved over to the left. But it doesn't have any left side, right? You're just looking at something that looks like the right side of a bell curve, and the left side is gone. Why is that? Because when people give statistics, most of the time in the US, these methods don't give any credibility to the hypothesis that maybe the guy isn't in there. And so they won't give a negative number. They won't give, like, the negative would correspond, like a -1 is 1/10. So the weight of evidence is always in favor of inclusion. So looking at this curve as scientists and forgetting everything else, like where it comes from, this is an interesting looking curve because this is a truncated normal distribution. The left half is missing because it's not allowed to be there. You're not allowed to have anything less than 1 whose logarithm is negative. It's centered at 0. This is if you fit it mathematically, the center of this half of a bell curve is 0, and it has the same tail on the right side as the blue curve. And so you can ask the question, what am I looking at here? Is this just some random number generator that has 0 information in it that is only allowed to tell me about positive events and throw out anything that might disagree with me? Well, that's a hypothesis we can test. And we did. So let me explain what this picture means. I'm going to draw a scatter plot in here in a second. On the x-axis is the actual information, which is obtained by the computer trying out 100,000 different solutions over a day. And that this is the best estimate that we have of what the information is. That's on the x-axis. On the y-axis is the reported information. Again, we're counting the number of zeros. 100th, 10, 1, 10, 100, 1,000. Same thing going up here. 1, 10, 100, 1,000. So that's the number of zeros. If all the information was present from human review that was in the information, then all of the, if you took any particular experiment and plotted it, it would go along this line, right? It would be right along there, because whatever the number was, it would be the same as information as it was by human review, give you the same number. If it's down in this region, the human number is less. If it's up here, if you put both, have left both numbers, if the point's up here, so that the computer interpretation is low, but the human interpretation is high, then people are overstating. And over here, it's just sort of imaginative that there's actually no information, or information the other way. And people are possibly really overstating it. So again, for every one of the points you're about to see, 2 different interpretations are made. One by computer to measure the information. You'll see that as a number on the x-axis. And also by people from reported cases. And what we see is that all these points don't line up along that orange 45-degree line. Right? Does this look like a really high correlation between a reported match statistic and the underlying information? Show of hands. Is this really high correlation? Okay, who thinks there's low correlation? Who's awake? Well, about 3/4 of the time, we're under the line, so people are being conservative. But 25% of the time, it's over or to the left, and people are not being conservative. And you can measure the correlation. So, like one of these points, so like this point over here, it's a 1 and a 1. That's along the line. The computer said it's statistics 10. A person said it's 10. But over here, the computer's saying that it's more like 1 in 100, but a person is saying it's 10. They're giving 1,000-fold excess in the match statistic. You can measure the correlation, and the correlation coefficient is 0.37. And what that means when you square it is you can account for how much of the match statistic that's used is explained by the information content, right? Presumably we're interested in identification information, right? That's why we're doing this. And since that number is about 15%, that means 85% of whatever CPI is doesn't have much to do with identification information. So that's the current state of human review. Huge DNA databases are built out of these methods of human review, where you threshold the data and you put them up onto databases. We'll talk about that in a second. But imagine you had a real DNA database that was really perfect in the sense that you try to preserve all the information. You took these genotypes and their probabilities as evidence from hundreds of thousands of cases. And then you took 10 million reference profiles, say, from convicted offenders. We'll hear how big that number is going to be this afternoon with arrestees and so on. And then the computer made a comparison that was just like an information comparison that computers make on a case-by-case basis. And it said, well, you know, that uncertain genotype together with, from evidence together with that reference has about 18 zeros in it. And so we think there's really a strong association. That's how you imagine a DNA database would work. It's an information system where all the evidence information is preserved and the comparison is giving you a information number. And if you had it, and this is all things that we've done with our true allele system at cybergenetics, you'd have an information preserving DNA database. You'd be able to solve cold cases with evidence to suspect matches, whether it was to implicate or exonerate someone. The science doesn't care which side you're on. Evidence to evidence cases to connect serial crimes. Kinship, evidence to kinship to find missing people. Evidence to reference for familial search, as we spoke about here 2 years ago at this meeting. And identifying victim remains, like we did for the World Trade Center for 18,000 victim remains. These are all things that information-preserving DNA databases can do. I spoke at the American Academy meeting last year about measurements that we did on this. And we took data from a 2009 validation paper that was 2 contributors. And we, in this case, we did it with victim known, victim unknown. But let me show you what happens. On this same x-axis of information that we've seen before, 10 would be 10 to the 10th, 1 followed by 10 zeros, which is 10 billion. 20 is some very big number. I guess it's 10 billion times 10 billion, whatever it is. That's why we count the number of zeros, right? And we measured what is the information against the true known genotype in the synthesized data that NIST made for us. And the answer is it's a sensitive method. We virtually always find the correct. So what does sensitive mean? It means that if the answer is true, you get the right guy. That's what it means, okay? And it, like the, I guess the Royal Canadian Mounties, you know, it always gets its man, right? It's got the right guy with an average of 17 or 18 zeros. Very sensitive method. Well, specificity is also important. How often do you get the wrong guy? And so we did an experiment where we compared with 1,000, actually now 10,000 random genotypes, and shown on the red is the exculpatory value. This is to what extent is this person excluded? And what you see is it doesn't go over 0. It's basically centered, very negative, with a number like negative 24. So that's 0.0000. It's the other way around. That's 12. So it's 1 in a trillion trillion. It's basically saying they're not there. So the methods that we're using either find the right people or don't find the wrong people. That's what you want from an information system. CODIS doesn't work that way. Instead, it takes these same threshold methods, draws the lines through the peaks. Half the time, people can't say anything at all. Half the time, they lose information. And when they get the information, as we'll see, most of the time, mixtures don't even end up on CODIS. What they do is they take these information-rich genotypes that they're not calculating, and instead just listing alleles. And they say, suppose there are 4 alleles at this locus, and you're comparing with this individual. So here we have evidence, but it's been very much cut down. Here we have an individual. So we're comparing between them. You take these 4 alleles, you take those 2 alleles, you ask, are they included? Losing information the way we just saw 10 minutes ago. But it doesn't calculate a number. It just says, is there a match? And it ends up finding hundreds of people. When it's only one person it's supposed to match from the evidence. And some of you in this room have experience when you use CODIS, you might get 100 names back. Well, either one of them's right or none of them's right, not 100. So this is very biased towards false positives. A lot of people are included. Why is that? Suppose the match statistic is of the data that they've put up that's all blurred out is about 1 in a million. Well, if you have 10 million people in your database, 10 people are going to come back. And 9 of them can't be right. We did an information comparison. You can see the talk. It's on our website if you're interested. We asked for the different types of databases, information preserving like TrueAllele or allele lists like CODIS, is it sensitive? Well, the average match statistic is around 15 zeros. And with CODIS, of the half of the mixtures you can even begin to upload, you can't upload them a third of the time. And is it specific? Are you making mistakes with false hits? With TrueAllele, that rate tends to be with a very small number like 0.3, or it doesn't give much of a match statistic. It's less that, how often does it go over 0? About 1 in 10,000 times. With CODIS, depending on what rules you use, you can go up to 25%. Because of that, CODIS doesn't let people upload profiles. In fact, we did a study with a local lab, and about 80% of mixture evidence that they get to doesn't go up there. So if half of the mixtures aren't being interpreted, and 20% of those are being uploaded, we're getting maybe only 10% of our evidence from mixtures onto our national DNA database right now. In our imagination, we think everything's up there. But that's just our imagination. Okay, so this is a case that the next speaker, Dr. Greg Hain-Pickian, asked Cybergenetics to look at. It was in the Western United States, and it involved a man who was accused of taking a gun, pointing it at 2 police officers, thinking the better of it, and running the other way. After being shot in the back with some number of bullets, he miraculously survived. The weapon he was said to have used was found thrown 35 feet away. They said he dropped it as he ran. And they scanned it for touch DNA, and the lab report said it was inconclusive. Well, TrueAllele took a look at it and found that, in fact, he was sort of excluded. It was uninformative, but if you put a match statistic, it's 10 to 1 that he's not in there. That's what the -1 means, 0.1. But we also found that there was another individual in there with 9. So there's a way, won't go into it, with information theory that you can say, what's the expected match statistic without making any comparison? How informative is this profile? The answer is, in this case, 9 zeros, billion to 1, billion. Okay, we tried getting it into the CODIS cut-down format where you slice and dice and you get rid of all the information you possibly can, and when it was done, it didn't satisfy their upload criteria, like the other 90% of the mixtures that never get in there. But we're looking at something where if we had this state's database, we would predict a billion match statistic, and it wouldn't find anybody else. So why can't somebody, like a judge, order a comparison be made using a newer technology? Since CODIS can't do it, and there are alternatives, why isn't information being used to exonerate people? He's still sitting in jail. From what Greg said last night, I think he's going to be there a very long time. This is a different case that we worked on. This is from Louisiana. This was a case that was an almost exoneration, where an individual was in jail for quite a decade or two, having been accused of a rape. They found some evidence that had been abandoned. As you know, a lot of DNA evidence is never used. This was from a rape kit up in an attic. And they wanted to test it. The reason is that ABO blood typing had shown that he wasn't in some other sample, and as Barry Scheck from the Innocence Project said, they have ironclad proof that this is exculpatory evidence. So to confirm that, they did some DNA testing, but it didn't confirm it at all. In fact, Trulio found about a 10,000 match statistic that said that he was on this piece of evidence. And a nicer sample went out to an independent defense lab, and they got an even nicer statistic. So he's still in jail. But this sort of evidence, whether it's locked up in attics, or it's underinterpreted and called inconclusive, or never sent up to DNA databases, is never taken from rape kits most of the time and compared against databases of suspect genotypes in order to find some true perpetrator. So DNA database identification with most evidence now doesn't happen. It's not even attempted. All right. So you have this concept, as we spend billions of dollars on DNA technology for law enforcement, exoneration, post-conviction, all sorts of things. I mean, don't you think that everything that could be done is being done for all that money? Well, maybe from some perspective using ancient 20th century technology. But in fact, from an information perspective, another way of looking at it is the government has a monopoly. They own the evidence. They own the DNA databases. They own the offender genotypes. They own the DNA axis. They own all use of their technology, and it's not really very effective. I mean, it is effective. It does find people every now and then. And there's so much crime. If you're finding it 10% of the time, that's wonderful. But if you're going to see somebody about some miracle cure, and they told you, we can cure a disease 10% of the time, if it's a life-threatening disease like violent crime, maybe you wouldn't be so excited about the 90% that wasn't being addressed. So, as we'll be talking about for the rest of the day, how do you balance public safety versus personal privacy? If you have anonymized DNA databases, which is what government uses, so we don't know who any of these people are, should law enforcement or defenders or post-conviction lawyers have access to being able to use better 21st century technology that's actually informative, that might be able to find true perpetrators? Okay. Here is one sort of project that we've been talking about. Imagine that you had a real information preserving DNA database. And there's some groups that we're discussing this with around the country and in other countries. So all the evidence, 100% of it, gets interpreted. Mixtures, no matter how complex. Whatever the evidence is trying to objectively tell you, it goes up into a database. It's there. There's no restriction on upload because you don't have a false positive problem. If it's going to match something, then you'll get some information number. And then you put likely offenders into the other half of the database, and you let it match. Well, suppose, I forget the exact number, it's somewhere between 10 and 50% of the number of women in the military who are raped during their service. It's a huge number. It's been in the paper a lot. There's also a problem with prison rape. We read in the paper about rape on college campuses. But imagine if you actually had a system that, instead of being designed to throw out most of the information, was highly effective in detecting rape. As if the evidence was collected and processed in a lab and uploaded onto a DNA database, and the rapist was in the database, there was no blocking of the uploads, and the information was preserved, and that one person would be found. What would happen in a closed population, like the Army or in a prison, where you could put all the potential offenders, like the whole prison population, say, onto a database, and every crime would be worked up? You'd find a lot of matches. And what would happen is the odds that a rapist would be caught would be a heck of a lot higher than it is today. And if people know they're going to be caught, maybe the incidence of crimes like this would go down. Because if you know with much more certainty that you are going to be caught. And if you're a serial rapist, you're going to be caught with a database like this. Then maybe for closed communities like the Army, it would make sense to put something in place as a DNA surveillance to prevent these crimes that are plaguing these institutions. Okay, well, thank you for your attention, and you're going to be enjoying, I was going to say subjected to, but enjoying another full day of maybe another 10 speakers or so. And on Cybergenetics website, as many of you are learning in your classes, we have a lot of material of courses, articles, presentations, and so on. And I just wanted to bring in a scientist's perspective on the information that's in DNA evidence as you think about some of the public policies that may not even be aware that there's an issue. Thank you.